A very warm welcome to all the joinees for today's session on this very exciting topic on lysosomal storage disorders. We know how the storage disorders are actually impacting our population, Indian population, especially at this moment. So I think this is a very important topic to understand how we can screen them, how finally biochemical understanding, what are the biochemical tests which are available in India, and how we can early detect these children to start off their treatment as early as possible. So I think but this is going to be a very, very exciting topic, and our most the most important part of this webinar is our expert, who is Dr. Anikha Bellad. It gives me an immense pleasure to introduce our distinguished speaker for today. She's not only a physician but a scientist and a clinical biochemist whose work spans from multi-omics research in rare genetic disorders. She holds an MD in my biochemistry and a PhD in molecular medicine. And currently, she is serving as a faculty scientist at the Institute of Bioinformatics, Bangalore, and adjunct faculty at the Manipal Academy of Higher Education. Dr. Vellard has had the distinguished experience of serving as a visiting scientist in the Department of Laboratory Medicine and Pathology at the world renowned Mayo Clinic in Rochester, Minnesota, USA, where she received advanced training in omics, technology, and rare diagnostics. Her research focuses on the development of new assays for diagnosis, monitoring, and management of the lysosomal storage disorders, and she has contributed to multiple DBT and ICMR funded projects, and her impactful publication spans from molecular genetics, proteomics, neurology, and clinical biochemistry. Please join me in warmly welcoming Dr. Anikha Bellad. Dr. Bellad, the floor is all yours.
Thank you, Dr. Upasana, for such an extensive introduction. Shall I say share my screen, Chaitanya? I'll be sharing my screen. Please confirm if my screen is visible.
Yes, ma'am, it is. It is. We can see your screen, ma'am.
Yeah, just a minute. Okay. Good evening, everyone. I'm Dr. Anikha Bellad, and I'll be presenting today on one of the most important current burden of the disorder, where lysosomal storage disorders, unmasking the silent burden through biochemical diagnosis. So understanding this has become a very important field. And as the audience here is multidisciplinary, so I have designed my workflow both clinically as well as to understand it more clinically as well as biochemically. So to start with, so as we all know, lysosomal storage disorders are, even though we see them rarely, but then individually they are rare, but considering like completely as a whole, they would create an extensive burden for the society. So today's talk will be focused towards understanding LSDs, a brief about it, and then followed by an importance of biochemical diagnosis and how it would complement genomic or the biochemical first testing or genome first testing, an emerging role of advanced technologies in understanding or diagnosing these disorders, and as well as therapeutic monitoring of these disorders, and followed by challenges and opportunities in India currently, where we can focus on rare disease diagnosis. So why this topic matters? As I was saying, lysosomal disorders collectively affects millions worldwide. even though they are individually rare, they are mostly are progressive and multisystemic. So most of the times what happens, they will be misdiagnosed or they are missed because of these multi-system involvement. The diagnostic delays would is reported to be in years, and diagnostic delays would contribute to irreversible damage for different organs. And early diagnosis can significantly improve the outcomes considering the emerging therapies in these disorders. And also, post national policy for rare disease, this has become as an important topic of exploration because the treatment is also available for most people through ORDI, and also need for integrated biochemical and genetic diagnostics in India. So early diagnosis is not just the diagnostic success, it also changes the outcomes and the life of many children. So coming to just to briefly to say the historical part of how these LSDs were identified. Even though the first phenotype of LSD was described early in 19th century, it is not until 1955 or 1956 when Christian Dew identified the lysosome physically. So, unless this time, even though the phenotype was discovered, the diagnosis was not the key. And then, followed by that, in 1963 to in 1960s, the identification of these were attributed to enzyme defects causing most LSDs. In 1980s, then they identified the molecular basis of LSDs, or the first pathophysiology was identified, and then the first enzyme replacement therapy in humans was given in 1990. And in the modern era, there are development of novel therapeutic approaches, such as substrate reduction therapy, pharmacological chaperones, and gene therapy. Alongside, there are also transcriptional control of lysosomal biogenesis, and the improved characterization of lysosomal biology has been studied. So what are the lysosomal storage disorder contribute actually? So how the lysosomal disorders are there are close to 70 rare genetic disorders. They result from a deficiency in a specific lysosomal enzymes or the transport proteins, leading to a failure in recycling macromolecules. So most of these are autosomal recessive disorder, except for Fabry, which is an X-linked disorder. And also the most common, these are monogenic disorder, that is a single enzyme is most often involved in most of these disorders. So it's very easy, like if you see biochemical pathways, it would be an ideal way to pinpoint these markers for the early diagnosis. So if there is a defect in any of these enzymes or the transporters, the undegraded substrates build up within the lysosome, causing cellular swelling. So as you can see here, in a typical normal lysosome, the different macromolecules like proteins, lipids, and carbohydrates are hydrolyzed, and a lysosome contains around 50 to 60 different hydrolases, which will degrade these macromolecules into the subset of the amino acids, free fatty acids, and sugars, and which are efficiently recycled. This happens in a normal condition. But what happens if there is a defect in any of the enzyme or deficiency or non-functional enzyme or a transport protein? So these would accumulate. Basically, the lipids, proteins, carbohydrates are not degraded and they accumulate in the lysosome, and this accumulated storage material creates a storage effect. That means that will causes the bulging of the lysosome uh, and thinning of that, and then the cellular swelling and inflammation, which will lead to the tissue damage or different organ damage depending upon which organ is involved. For example, uh, in Gaucher, if you can find, there will be accumulated lipids or sphingolipids, and if there is an accumulation of glycosaminoglycans, it leads to MPS, mucopolysaccharidosis type of disorders. And if there are oligosaccharide defects, then oligosaccharide accumulation would happen, leading to Pompe. So we all call lysosomal storage disorder as a rare disease, but are they really rare? So if we see the collective burden of LSDs, it is estimated that 1 in 5,000 live births, the estimated global incidence of LSDs is seen. And in India, currently there are more than 12,000 reported patients, but the estimated total burden is likely to be higher, 10 times higher than the reported ones, because of underreporting and missing numbers. So, and 30% of them have been linked to a consanguinity rate in our country. So, LSDs account for significant pediatric mortality and lifelong morbidity across the Indian subcontinent, which is still a silent burden because it's not been a major hike yet. So, the diagnosed population is only the tip of the iceberg. So, when we put all this LSDs into the bin of the inborn errors of metabolism, which are common inborn errors of metabolism, such as intermediary metabolism defects like urea cycle disorders and organic acidemias, you can say 30% of all biochemically diagnosed IEMs are LSDs. So it's a huge burden. And if you can see in Indian population, the frequency reported is Gaucher followed by MPS type of disorders and other glycogen storage disorders. So this is one of the studies which was reported in Lancet, where you can see the burden of LSDs in India, and as we were saying, almost half or 50% were close to the defects in the degradation of glycolipids, which includes oleosphingolipidosis, followed by mucopolysaccharidosis, and followed by defects in degradation of sulfatides like leukodystrophies. So considering this, if you consider this, so even if you want to diagnose LSDs, the major part, so you need a ideal tool that could multiplex these assays as a part of a newborn screening, where you can identify uh, these disorders early on. So there is a need for a biochemical testing where it can be diagnosed in early stages. So why LSDs are commonly missed? Because of the varied heterogenic presentation of these disorders, and they are progressive in onset. So many a times what happens, they are missed, they are settled at the early, like when the child is born, so they may be missed. And then as the child starts to progress, then because of the multi-system involvement, again, they are misdiagnosed many a times. And they will do shopping from one physician to the other physicians because of undiagnosis. And then the limited awareness. And if you see in a career of a physician, only few LSDs they would see, or some people they may not even see any of the LSDs. So the awareness, because it's not being seen regularly, that is not the first diagnosis of choice. So that's the reason most of the LSDs are missed, and restricted access to specialized testing. Especially these all studies are from a specific region, and there is no access to the testing for the rural, especially, and these are the specialized testing is limited to metropolitan cities. So that is one of the reasons why LSDs are not reported highly. So seeing the spectrum of the disorder would start as the one end of the spectrum has a classical infantile type that would present early on in the life, that is less than 1 year, which is mostly severe and rapidly progressive, and which involves a significant neurovisceral involvement. On the other hand, you have an adult, on the extreme end, you have an adult or attenuated form, which is a late onset, which usually starts in the teenagers. This is the one group where you would often misdiagnose as routine cardiac or renal issues. So as this presents, because there will be a residual enzyme activity still present in these cases, their presentation would be mild, and also they would present as a multisystemic disorders, and then that's why they are often misdiagnosed to have some routine cardiac or renal issues. So even with the heterogeneity is so high that even with a similar genotype, there would be multiple different phenotypes. So that is also one of the reason why do we miss or why do we confuse with the LSDs. And in between, you have a subacute form or a juvenile form, which are intermediate onset, slower progression, and manageable symptoms, but leads to irreversible organ damage without therapy. So this is a larger spectrum of disorder which starts from the early onset, that is infantile, towards the adult phenotype. So each of these disorder has a infantile phenotype for these, and then with the multisystemic manifestations, and you have a juvenile type and also late onset type or adolescent, and then you have an adult type. So even though most of these LSD have an overlapping symptoms, certain LSDs tend to have a particular organ which is more preponderance to. So for example, in Gaucher, mostly you have a neurological involvement, and also in cases of Pompe, if you go, there would be more of cardiac or a muscle involvement, basically. So any cardiac muscles or a skeletal muscle involvement. In case of MPS, as you all know, it would always mostly present as a skeletal manifestations. So even though there are some preponderance, but largely on the other hand, they are mostly are overlapping symptoms. The global developmental delay would be seen in most of these cases. So yeah, there was one study in the Journal of United Metabolic Molecular Diagnostic Reports, which says they had segregated LSDs based on the organ involvement or the system involvement. They had reported 307 cases where they found that 45, that is close to half, 50% of cases had of the Gaucher had hepatosplenomegaly. So and then in the Pompe, as I was mentioning, they mostly are muscle involvement. Close to 20% had muscle involvement like cardiomyopathies, hypertonia, delayed motor milestones, and most of the MPS group of disorders, mucopolysaccharidosis group of disorder, had skeletal involvement. And certain Niemann-Pick disease and other things, you might have ophthalmological involvement where you say you have cherry red spots and other things. So that would tell us about it. All these LSDs are mostly multisystemic involvement. So this slide will actually tell you how much there is a diagnostic dilemma and there is a diagnostic delay. And this actually this illustrates how an LSD patient, most of the LSD patients would start their journey and then till from the development of the symptom till the diagnosis, they would almost take 4 to 5 years for the diagnosis. This is not just in India, this is globally. So the timeline or the time lapse between the onset of the symptom to the diagnosis, it takes 4.7 years on average. And in India, it might be still more. So what is the cause for this? So when the child is born, most of the babies would be normal when they are born, but then with a mild symptoms or mild signs and symptoms, which are often subtle. So, but you should be careful about the red flag signs are the neuroregression, unexplained organomegaly. If a child has an unexplained organomegaly, or there are any skeletal changes or ocular sign, these are the red flags. If these red flags are missed, even though they are milder and subtle, so mostly there are tendency to be missed. So the first clinical contact happens from 0 to 6 months when the mother starts finding some abnormality or some difference than the normal children. So there might be a developmental delay, which the developmental milestone delay, which is the initial thing the mother may come up with. So at this stage, what happens when they would initially go to a primary physician or a pediatrician. So as these are the common signs and symptoms for most of the pediatric, the first diagnosis would be attributed to cerebral palsy or some of the nutritional defects or some of the other common neurological disorders. So then starts the odyssey of misdiagnosis from 6 months to 4 years as the child grows on. So at the first stage, the child disease will be biologically still active, but then the symptoms and signs would be subtle. And then as the signs and symptoms would increase, the parents would take the child to different specialists like orthopedists, gastroenterologists, or a neurologist. And then the battery of examination starts. So meantime, they would establish a baseline biomarker, or the biomarkers would evolve, but then there would be this is actually the phase where there is a real odyssey or real diagnostic dilemma, because there will be multiple diagnosis and then there will be a fragmented evaluation by different specialists, but still the diagnosis is not found. And then, however, you have to remember the disease is still progressing, and then the family stress increases. So this is when at some point they would refer to a metabolic specialist or a geneticist, which would almost take 4 to 4.7 years on average when the child reaches here. So this is when the first time a geneticist or a metabolic specialist would suspect inborn error of metabolism. So then again, depending upon the symptoms and signs, they would order different enzyme assays or DBS or leukocytes from the leukocytes, and the unmasking starts. So from here, this is the years of uncertainty would be actually is solved just by a simple enzyme assay, which is a biochemical investigation. And then you go for confirmation by using the genetic testing. But again, most of the 60% of genetic testing would come as variants of uncertain significance. So again, genetic testing alone would not be sufficient enough to identify whether the patient is really sick or that is the pathogenic variant which is causing the disorder. So during these 4.7 years, what happens to the child? So there will be continued evaluation or continued damage to the organs, and in some of them, they already cause the irreversible damage. So this time is very critical to diagnose and treat, because treatment currently there are enzyme replacement therapy and substrate reduction therapy for many of these LSDs, or at least they are upcoming. So, and even though they are available, so the timeline during which you treat them is also matters. So this is where you need a test or a diagnostic platform where you can identify this, reduce this 4.7 years to days or weeks. So ideally, it should be the day one, the child presence, and then within 1 to 7 days, you do an NT2 screening or the first line assays, where you do either enzyme assays or some of the mass spec, whether it can be a fluorometric assay or other platform assays, and then you do a biochemical confirmation by quantitative enzyme assays and find a deficient level and confirm by gene sequencing or targeted sequencing, and then establish a baseline marker, and then you have a treatment. You can plan for your treatment. So the current approach to clinical diagnosis is, as you can see, this is a very complex workflow. And then even though it starts from the suspicion, clinical suspicion is the major which drives the clinical diagnosis. So once there is a suspicion, then the clinician would go on. It's a stepwise approach, basically. So you will do the first line assays are enzymatic assays, where you can find, even though there is a limited panel for enzyme assays, not all LSDs have an enzyme assay currently to identify. And then there are other assays like urinary glycosaminoglycan for MPS, and urinary tetrasaccharide, glucose tetrasaccharide for finding the oligos, which is based on electrophoresis, or mostly based on the electrophoresis, like you will have other things. And then you also have, then you go for the confirmation by genetic testing. So, but what happened in the post-genomic era? When you go to a clinical geneticist, they will not usually, they will usually go it in a reverse direction. So the first thing they do would be the gene panel or exome or genome sequencing. So in the post-genomic era, many of the clinicians are opting for genome first approach, or even though if they don't do whole exome or whole genome, you do a targeted testing, the targeted panel sequencing, and then based on that, you have an unknown pathogenic variant or a known pathogenic variant, and then confirmation by biochemical testing. So either way, patients do where the patient land up. And again, these enzyme assays or these genetic testing, everything is mostly available in the urban cities. Again, rural are underserved. So again, there is a lack in the diagnosis there. So what are the challenges in the conventional assays? So as we know, the first biochemical or the first line of diagnostic assay is enzymatic assay, which can be done by different platforms. It can be a fluorometric assay or a mass spectrometry based multiplexed assay. And you can use DBS or the peripheral blood mononucleoside, that is leukocyte assays. And also you have a functional evidence. This biochemical assays mostly provide you the functional evidence of the disease, and also it will tell you about the rapid progression. And post-therapy, how the patient is responding. That also you can do, even though enzyme assays would not help you much there. So, but what are the challenges in these conventional assays? One is the limited specificity, limited availability, and also you need to do a single analyte testing each time. And the major problem with the enzyme assays is pseudodeficiency. That is, in vitro, you can have false positive or false deficiency of the enzyme, but the child may not actually have it. So residual activity overlap, and there are sample instability issues, and also assess standardization across laboratories. So challenges on the contrary, the challenges to the genetic testing, the major blind spot is the variance of uncertain significance. So this is the major burden, and as I mentioned, it is reported that 60% of these cases would end up in variance of uncertain significance. That is, again, you are in a dilemma whether a child actually has the disease, whether the child that has the disease is actually due to that particular variant. So, and then you have a poor genotype phenotype correlation, and the problem of novel mutations, and therefore there is a need for biochemical correlation. Always biochemical confirmation is necessary, especially to understand the progression of the disease as well as um, the functional evidence of the disease. So the integrated diagnostic workflow is the best one. The first one is a clinical suspicion, followed by screening by dried blood spots for first-line enzyme activity assays, confirmatory assay by leukocyte to fibroblast enzyme activity measurement, and molecular testing, genetic sequencing to identify specific mutation, followed by a family counseling. But again, this is still a timely approach, and then again, still it takes the different step. It's a multi-step approach. So currently, the RUSP has also included, that is the it has included in the newborn screening for Pompe disease, MPS 1, 2, and infantile Krabbe disease, even though there is no universal screening program in India currently. So this is a recommended universal screening panel recommended by US thing. So moving on to the newer markers, which are emerging markers in LSDs and which can change the diagnostic standpoint at this for the LSD. So biochemical evaluation is the key in the diagnosis of LSDs, from conventional assays to emerging markers in LSD. So these markers would aid in diagnosis, monitor disease progression, as well as assess the treatment response. So why we need a biochemical testing? Because these are all dependent upon the accumulated substrate. So you have a pathway. If you take a pathway approach, which can be multiplexed, for example, I will not go into biochemistry of these because it would be too extensive and too boring for many of you. And so this is basically to say how complex are the pathways, and based on this, there is a I would be talking about mostly the Gaucher, Fabry, and Niemann-Pick because those are the common disorders which are being reported in Indian context. So if you see the sphingolipidosis pathway, instead of going for individual enzyme assays, if you have a multiplexed panel which would detect all of these pathway substrate or the metabolites in a single assay, which are related to a particular disease. For example, beta glucocerebrosidase deficiency would cause a Gaucher disease. The alpha glucosidase would cause a Fabry disease. So if you are measuring ceramides and the acetylated form of those are lyso-Gp3, if you are measuring all these in a different thing in a single panel, and if you could identify all these disorders in a single assay, how that would help. So next is the mucopolysaccharidosis. You have different glycosaminoglycans accumulated in these cases. So that would cause you, leave you with a different uh, MPS disorders. The third one is the oligosaccharidosis, which is your Pompe. So where glycogen defect or the degradation of the glycogen would be affected, and then there will be an accumulation of the glycogen will not degrade in a proper manner. So there will be accumulation of oligosaccharides. So you will measure the glucose tetrasaccharides. And if you can see this is the entire Pompe thing. And then there are other minor disorders which are quite rare. So as I was talking about the sphingolipidosis in a bigger picture. So if I talk about Gaucher disease, this will convert glucosyl ceramide to your ceramide. So if this enzyme is defective, then what happens to the ceramide? What happens to the substrate? And what happens to the product? So if you're measuring these two, or if you're measuring the ratio of these two, that would give you a better picture of the functionality of the enzyme. As well as if you're talking about leukodystrophy, metachromatic leukodystrophy, the sulfatides as urinary sulfatides are the markers for these disorders. So you can see there would be this accumulation. So similarly, you have like for the Fabry. So you have lyso-Gp3, which is the accumulated substrate, and then what happens to the product. So what is the ratio of these two? That would give you the functionality of the enzyme. So similarly, as I was talking about the Pompe disease. So alpha galactosidase enzyme defect, the glycogen basically will break down into glucose, pyruvate, and lactate. But if there is a defect in this enzyme and then the glycogen is not degraded and it goes to lysosome where this is not gets degraded, and then it actually this particular enzyme would help degrade completely, and if there is deficient of this, then there will be an accumulation of oligosaccharides which are degraded at different lengths. So this is again the complex pathway for your glycosaminoglycan degradation pathway. So if you are able to detect in a single assay, for example, all types of dermatan sulfates different during this different pathway, you can identify at least these MPS. And then if you have an assay which can detect all your chondroitin sulfates, you will have different MPS disorders to cover. So coming to an integral membrane protein disorder, which is a Niemann-Pick type C disorder. So if you have a Niemann-Pick type C, it's an integral membrane protein. If there is a defect in that, there will be a cholesterol accumulation, and then the ketocolesterols are the markers for your disease. So we have developed some of these assays. These are the novel assays because I'm working as a scientist as well, and we have developed many of these multiplexed LC-MS based assays which are ready for clinical translation. So based on those targeted multiplexed assay, we have solved a couple of cases. So I'll be talking about three case scenarios which will help you to understand how the biochemical investigation would actually help you to functionally validate the or reclassify your VUS. So we had a case of a 7-month-old male child born to a third-degree consanguineous parents. They presented with mild global developmental delay in spastic quadriparesis. Birth weight was 2.2 kg and APGAR score was 7 with a history of cord around the neck. So on examination, they had microcephaly, coarse facial features with dysmorphism. There was an hemangioma over the forehead, stridor nystagmus, cortical thumb, and spastic quadriparesis with limb hypertonia. Basically, to say there was some of neurological involvement as well as some skeletal involvement, like musculoskeletal involvement. And then the clinical whole exome sequencing was performed by the clinician, by the geneticist, which found a homozygous missense variant in DEGES1, which was classified as VUS, variant of uncertain significance, as per the ACMG criteria. So the clinician was, the clinical picture was not completely resembling, like not a textbook picture of LSD they were suspecting about, but then it also showed a variant of uncertain significance. So the clinician was not sure. So they asked us whether we can identify some of the substrates accumulated and they can identify whether the DEGES1 is the disease causing variant in this particular case. So similarly, there was from the same clinician, but it's a different patient, not from the same family. So there was a 2-year-old male child presented with global developmental delay with spastic quadriparesis, but there was no parental consanguinity in the child reported, like in the parents. The neuroimaging revealed cerebral atrophy and thinning of the corpus callosum with features suggestive of leukodystrophy. So this suggested the leukodystrophy. At presentation, a clinical suspicion of Lee syndrome or mitochondrial leukodystrophy was considered. Initially, the clinician thought it is a Lee syndrome. There was a family history was significant, like father affected with dilated cardiomyopathy and sensory neural hearing loss, suggestive of some kind of genetic variation present. So clinical whole exome revealed homozygous missense variant in again a DEGES1 gene at a position of 255 where aspargine was converted to serine. So this was reported as a pathogenic parent as per the ICMG criteria. So here, even though the clinicians, there was a differential diagnosis of mitochondrial leukodystrophy. They identified based on the reported cases, they were suspecting DEGES1. So these two cases we got from them to identify which are the markers which we could identify. So what we did was we performed the pathway approach and covered the ceramide synthesis pathway metabolite. So you can see this here is the DEGES1, which is a desaturase, which converts dihydroceramides to ceramide. This is a ceramide synthesis pathway. So the dihydroceramide conversion to ceramide was affected, leading to the disorder caused by DEGES1 is hypomyelinating leukodystrophy. So if you can see this picture, there should be an accumulation of dihydroceramide in this case, and there should be a decrease in the ceramide levels in these two children. So what will happen? But we were curious to know whether the VUS case, we could reclassify it, or basically to identify whether it is a pathogenic thing. So to identify that, we went to and checked for what is, like, you can see where the variant is, whether it is in the critical region of the gene. So the guess one is on the chromosome 1. So, and the first variant, that is the VUS, sorry, I think it's mislabeled here. So the first variant, which is VUS, was in the exon 2, and then the second variant, that is pathogenic variant, was also in the similar, like it, yeah, that's in the exon 3. So that these two, but they were both reported in the fatty acid desaturase domain, that is a functional domain of the gene. So functional domain of the protein. So if you see the protein here, so that was in the functional domain of the protein. So it should be causing the disease. So when we went to see whether, because this was not reported in any of the ClinVar or any nomad thing, so that's why we wanted to understand how this variant is. So we looked at the evolutionary conservation of this residue. If you can see, it was evolutionary conserved. So most of the time, it's not always, but at least most of the times when it is an evolutionary conserved, we think it is an important amino acid at that position. So that's why it's evolutionary conserved. And if any defect in that, or if there is any variant, it would most probably cause a disease. So here at aspargine at 113 position is converted to isoleucine. Normally, there should be an aspargine which was converted into isoleucine. The other variant, which is already reported in a ClinVar as a pathogenic variant. So here at 255 position, so here also the aspargine is converted into CJ. So then we performed an LCMS based targeted assay for sphingolipidosis, considering all the sphingolipids in the pathway. So we measured all the different analytes in the sphingolipid, in the ceramide synthesis pathway. So dihydroceramide, ceramides, sphingomyelin 1, like sphingosine and sphingosine 1 phosphate, and also these deoxysphingosines and deoxyceramides. So you can see the defect is here, and you can see the two red dots are the patient samples. And then these are the control samples. So if you can see, the farther away is this one, that is, which you can see at 6 micromolar concentration. So this was the VUS case, and this was a pathogenic case. So you can see there is an accumulation of dihydroceramides, significant accumulation of dihydroceramides in this case. And then there is a decrease. On the right side, if you go, it is increased fold change. And then on the left side, if you go, it is a decreased concentration. So you can see the ceramide levels were decreased compared to the controls. So, and if you take the ratio of these, that would help you to understand and you can see whichever we were thinking as VUS was actually, there was accumulation of dihydroceramides in significant amount in that particular child. So that was one of, then we went back and reported that these are the cases, and then they would take it forward for further evaluation. So another case, one individual with no conclusive diagnosis by genome sequencing. They did a whole genome sequencing. There was no conclusive evidence, but with two VUS and GM2A gene. So which was paternally and maternally inherited. They found two VUSs and GM2A gene, but they were not sure whether this is the gene which they are looking for. No abnormal accumulation of CSF, GM2 gangliosides were identified, which is because of this gene. Fibroblast cells, then they procured fibroblast cells from the patient. We received patient-derived fibroblasts for ganglioside profiling. Targeted assay for glycosphingolipids, including gangliosides, were analyzed in our lab. So this is the ganglioside degradation pathway. So you can see GM1 gangliosides are converted to GM2 to GM3, and then lactosyl ceramides and to glucosyl ceramides. So, and then here what we have, the problem with these are GM2A protein. That is, GM2A activator protein was defective. As they were talking about GM2A gene. So this was the defective variant of the enzyme in this particular case. So when we did the profiling of these cases, if there is a defect in the GM2AP or GM2A activator protein, the GM1 would accumulate, and then your GM3, the conversion, because GM2A is involved in conversion of GM1 to GM3 along the way. So GM2, so GM3, we identify GM3 as a reduced concentration, and then GM1 and GM2 were accumulated in these cases. So we could identify but still it was a single case. But at least at the biochemical level, we could identify there is an accumulation of the substrate. Sorry, just a minute. I'll just on my. Sorry. So how about other disorders in this pathway? Because we were talking about gangliosidosis, we had another we had another 3 cases, which two were Tay-Sachs and one was Sandoff. And if you can see here in this pathway, so GM2 to GM3 and GA2 to lactosyl ceramide causes Sandoff, and if there is a defect in beta hexosaminidase B, that is hex P, if there is a defect, then Sandoff's is caused, and then if hex A is defective, Tay-Sachs is caused. So in hex A, you should find the accumulation of GM2 and then GA2 in cases of Sandoff. So if you can see here, GM1 is converted to GM2, GM2 is converted into GM3, and then GM3 to lactosyl ceramide to GA, and then GA2 is converted to lactosyl ceramide. So Sandoff's, if you see in this particular panel of analytes, the first panel is the control, the second is the Tay-Sachs, the third is the Sandoff. So in Sandoff, there is an accumulation of GA2 substrate. And in cases of Tay-Sachs, so where GM2 has to convert into GM3, so there is an accumulation of GM2. So these are basically the markers which can be used, and these are pathway approach. So multiple analytes can be clubbed as a single pathway, and then you can identify different disorders in a single run. So this is for an Niemann-Pick disease where the marker for Niemann-Pick disease is sphingomyelin, and for the Gaucher, which is the most common we were talking about, is the glycosylsphingosine, because the beta-galactosyl ceramide is defective. The glucosyl ceramide is not converted to ceramide, so that it takes an alternate pathway where it will undergo for glucosylsphingosine. So you can see there is an accumulation of glucosylsphingosine compared to controls. And so this is a case of Fabry. We had 6 cases of Fabry. You can see they were of different variants. You can see the levels of lyso-Gp3 accumulation. And this here you can, and this is the normal range, which 0.08 nanomolar, and then the fold change. It can be as high as 1,000 fold change in a particular case. So this were the difference in the lyso-Gp3. There was accumulation of lyso-Gp3 in cases of Fabry disease. So this will tell you about how these metabolites can be used for early diagnosis. And also, this is a Pompeii where glucose tetrasaccharides are accumulated in the probands. So you can see the accumulation of glucose tetrasaccharides in the proband based on the LCMSMS based assay. So currently we have developed these LCMS based targeted assays. So these are the different, you can see the different type of LSDs and a defective protein or enzyme. And these are the substrates which we are measuring, and the substrate and products which are measured using the LCMS method and corresponding to a certain class of analytes and a disorder. So this is one of, these are for sphingolipidosis, and these are for MPS types of disorders, and this is a mucolipidosis type where you have a GNPT. N-acetyl glucosaminal phosphotransferase catalytic subunit would be defective. So again, there will be an accumulation of oligosaccharides. And then a glycogen storage disorder, which we were talking about, Pompe. And then you have NPC1 or the Niemann-Pick type C, where we have the problem with the cholesterol transport and then the accumulation of 7-ketocholesterol and other thiocholesterols which are accumulated, and then this would be measured by LCMS based targeted assays. So these two are these are the panels which we currently have developed, and we are using them for understanding the disease. So not apart from the diagnosis itself, you can also use these biomarkers for the treatment monitoring. So you can, at the 0 day or at the time of the treatment, the markers can be sky high. And from 4 to 12 weeks, there would be a rapid reduction of these markers. And this is the study which we are currently, it's ongoing. So we are taking patients from pre-ART and post-ART therapy. So which would help us to understand how these markers are useful in treatment monitoring. And it's already established also. So we have certain cases which are showing this trend. So these can be used as the thing. So currently the challenges or the unmasking that has to be done in India is one of the major hurdle is the economic barriers, the high cost of these tests, lack of universal insurance coverage of rare screening programs, and then the infrastructure. As I was saying, these were all limited to metropolitan hubs. And then there is an underrepresentation of rural population where the most of these IEMs are mostly common in the low economic group. So awareness, low clinical index of suspicion, the major problem, because there should be an awareness among the clinicians, physicians and pediatricians, especially the primary care physicians, to think of an LSD whenever there is a multisystemic disorder. Whenever there is a multisystem involvement, think metabolic. So, and then there is a lot of policy gaps where you need a mandatory newborn screening program for treatable LSDs at least, like Pompe or Cauchy, which are also a part of National Program for Rare Disease, which was released in 2021. So these are my references. And you should remember, biochemical diagnosis is no longer just confirmatory, it is becoming central to precision rare medicine, rare disease medicine. So thank you. I'm open for questions.
Thank you so much. Hello, I was actually speaking on mute. Hello.
Hello. Yeah, thank you.
Yeah, thank you so much for this very illustrative and informative presentation, ma'am. It was really wonderful. It just gave me the entire another time of brush up of lysosomal storage disorders. I think there are few questions. Quickly, we
Yes.
can address that if you're okay.
Yeah, I can. Should I go through them or you would read it?
I am just giving. Dr. Naresh Teyade is here, and he has asked if ERT for NPT available in India. And what would be the biomarkers testing cost?
The testing cost for these biomarkers is not uniform across the places. It is in thousands. Like we had actually made this exercise, and then we identified from different, it can be from 2000 to 10,000. So it varies a
Yes.
lot, and most of these biomarkers are not present in India, whatever we have developed, but we have a clinical grade assays currently. We would be launching them soon, but then currently, if anyone are open for these, you can send them as a collaborative uh, studies. Like we are taking collaborative samples where they're currently we are not costing them, but we are analyzing those samples. So but these, whatever I talked about, these biomarkers are not currently available in India, especially for lyso-Gb1, lyso-Gb3. Glucose tetrasaccharide is there, which is not a mass spec based assay, but an electrophoresis based assay. So yeah.
All right. Mr. Bishwajit Saha, he has actually given us certain information where these investigations inform methods, including cost, are done in India. Enzyme assays, DNA-based investigations, biomarkers, siblings, cost for patient siblings and for birth parents, how the samples are sent, including the stability and cold chain requirements. I think the DNA-based samples are sent for the EDTA blood.
Yeah.
Those are
That
normal proteins
Yes.
supplied here.
Yes.
And of course, the stability, as you mentioned, that matters for the enzyme activity assays, enzyme assays.
Yes.
Yeah. So,
So the Sorry.
yes,
Yeah, please.
no, yes, ma'am, go ahead, please.
The testing for and like DNA based investigation, it is done in most of the referral genomic labs like Medgenome and other Sandoff and many of the genomic labs, because these are the common genome panels which are available. Enzyme assays are done in certain laboratories like CTFT Hyderabad, and uh, Lalipath also has RV Metropolis has some of these assays. Even Medgenome has some of these assays. So uh, yeah, I have not shown the exact like which laboratory have these enzyme assays, but most of these. And again, for all the LSDs, we do not have enzyme assays. Only few has enzyme assays. Uh, and those enzyme assays are mostly in the referral laboratories. The cost again varies from lab to lab.
Yeah. Yeah. Thank you so much, ma'am. I think we don't have any such questions further. Okay, there is one question from Mr. Shiddesh. How can the newborn screening programs effectively differentiate between the infantile onset and the late onset forms of LSDs to minimize the uncertainty and clinical management and family counseling? I think this is a very important
Yeah.
question when we consider NBS.
Yes.
Yes.
This is a very good question, and in the newborn screening programs, mostly we have, because as the child has the subtle manifestations, you would find the markers as such in the infantile form. But in cases of late onset forms, what happens? The clinical suspicion is not there, but there is a residual enzyme activity, but it's not, still the enzyme activity is not at the fullest. So you can even identify some of these markers. I'm not saying all these markers would be present, because they accumulate across the time. Then only you will be able to identify them. So at least for the late onset time, it may not be possible everything you would identify them in the newborn thing, but some of those can be identified depending upon the accumulation of the substrate. So because this is a progressive disease, so the symptoms would
Yeah.
appear in the later stage of life, but at the early stages you can identify some of these markers in these forms of LSDs.
Correct.
But late onset, it is still difficult to identify at the newborn stage, because it takes time, because there is enzyme activity still. It takes some
Yeah.
time to become accumulated.
True. And that's I think, the challenge for the late onset, for the early detection there.
Yes,
And I
that
think,
yeah,
yeah,
that's why
I
we
think the
need to
process
be
in
aware
UK,
whenever there is a multisystemic disorder, then we need to think of a metabolic cause, and we can do, even in the late onset, we should plan to do some of these assays.
yeah. I think what is coming right now in terms of genetics is the newborn genetic screening. I
Yes.
think what UK has already started, and we have also started in India a couple of years back in a private sector. But I think that is the future, to understand the late onset, late infantile syndromes, and the pathogenic conditions which can actually affect in this way. Precision medicine ahead, I believe.
Yes, because again, genetics has its own thing. As I was saying, 60% of these are VUS. So again, there will be a dilemma of whether to treat them, whether to leave them. So again, it's always a complementary testing, both integrated workflows. And when it comes to
Yeah.
cost, especially for Indian scenario, biochemical would be the first line. And then, yes, definitely, even genetics can be a first line. But again, addressing the US would be difficult if it's a genetic alone.
True. Thank you so much. This was really very much insightful and a very richly informative presentation. And I think we end here today. And on behalf
I
of the
would,
organizing
yeah.
team and from the audience, we extend our heartfelt gratitude to you, and to our audiences also, who has kindly joined us for this webinar. I thank you, everyone, and hope to see you soon in the next session. Thank
I
you
would
so
like to
much.
thank ORDI for giving me an opportunity. It was really good to be with, yeah, thank you.